Um, okay, hello everyone. My name is Nishel Narajos, and I am a senior at Glendora High School, and my research is on the binding analysis of a poly-N acetylglucosamine-specific phage to structurally similar bacterial polysaccharides via an ELISA. My mentor was Shelby Vexler, who is a PhD student of chemistry at the University of California, Los Angeles. So firstly, I'd like to give an introduction to what is antibiotic resistance, or AR for short. So a definition for AR is the bacteria gaining the ability and evolving to survive exposure to antibiotics that would normally kill them and stop their growth. And ever since the first antibiotic was discovered in 1928, penicillin, antibiotics were administered to all illnesses, simple and severe. And this led to antibiotics over time evolving and gaining resistance to those treatments, and which has led to antibiotic resistance, posing as a significant public health challenge because it can lead to longer hospitalization stays and is overall emerging as a global health threat. And some statistics to back this up. Globally in 2019, it was found that there have been 1.2 million deaths that were directly linked to an antibiotic-resistant infection, and nearly 5 million deaths associated with AR. And with these staggering numbers, it is imperative that scientists and medical professionals find alternative methods to pick up the fight where antibiotics left off. And one of these treatments is called phage therapy, which is a type of treatment that uses the help of bacteriophages, which is unique to antibiotics, or called phages. And these phages sort of look like little spiders, as in the image to the left, but actually through a microscope, they look more like spaghetti noodles. And these phages are naturally occurring in the environment and in the human body, so they are not foreign to ourselves. And a positive is that they only attack bacteria and are pretty effective at this. And a cool thing about them is that they can undergo genetic and chemical modifications to overcome the two main limitations that come with phage therapy. With one being that phages are hyperspecific, meaning that phages can only bind to a very specific species of bacteria, thus limiting their host range and the bacteria that they can bind to. And two, not all phages necessarily lysis the host cell. And when you have a phage that is lysogenic, it means that it doesn't necessarily kill the host bacteria cell, and it leads to more production of a progeny phage and more bacteria, which isn't as effective at combating the bacterial infections. So the phage can be genetically and chemically modified to produce a more effective phage overall. And taking a M13 bacteriophage, which is the phage used in my study, um, the genetic modifications include focusing on the receptor binder protein, or the G3P protein. And while the receptor binding protein RBP of M13 is usually responsible for binding to the F. pillus of E. coli, RBP can be modified to bind to a common bacterial antigen instead, which is PNAG. And I'll be going over that more later. And since it has a bacterial polysaccharide that it's able to bind to, it can increase its uh, bacteria that it can bind to, and expanding its host range. The chemical modifications include the alteration of the major coat protein, the G8P protein, and these lead to the attachment of antimicrobial polymers as seen through the green polypeptide chains. And these can be very potent polymers that can lead to the lysis and destruction of bacterial cells. And with these two modifications leads to the overall SEFV phage that we studied. And this phage is a short chain variable fragment phage, and it has been genetically and chemically modified form of an M13 phage, and serves as a delivery platform for um, potent compounds, as seen by the green polypeptide chains. And moreover, the receptor binding proteins are modified to bind to poly-N-acetylglucosamine, or PNAG. And PNAG is a common surface polysaccharide that is found nearly 30 species of bacteria, including E. coli and Staphylococcus aureus. And this antigen can be expressed as a short chain variable fragment, which is fused to the M13's receptor binding proteins, which helps it direct the phage and uh, the polymers that's included to help target bacterial infections more effectively and specifically. So this leads to my research question: can the SCFV phage bind to carbohydrates structurally similar to PNAG, thus expanding the range of species for potential treatment? And the methods I took to researching this are in three parts. Would be firstly the preparation of the phage, then the purification, and finally the main experiment, which is the ELISA. And the phage preparation includes getting the two main phages that were studied, which is the wild type M13 phage and the SCFV modified phage. And that included the steps of inoculating autoclave media and incubating it with the M13 producing E. coli. And also for the FCFV phage, it was supplemented with ampicillin and with a helper phage. Now, for the phage purification step two, it was done to concentrate the phage and obtain a pure sample through filtration and the centrifuge of the media. So after finally getting a pure form of the phage, which leads to the main experiment, which is in ELISA. And we conducted an ELISA protocol, which is an enzyme linked immunoabsorbent assay, which studies the binding of the two types of phages to three different types of carbohydrates, which is chitin, chitosan, and cellulose. So here's a more detailed graphic of the ELISA. It was conducted on a 96 well plate, and the steps include having the binded antigen or the carbohydrates binded to the bottom well of the plate, and it is then incubated and blocked with a BSA blocking buffer, and this prevents any non-specific binding, which could lead to false positives in the final results of the step. So after blocking the antigen, it has another wash and incubation period in which the phage was added and to test if it binded to the surface antigen or not. And then another wash and incubation period to add the horseradish peroxidase antibody, and then adding the TMB solution, which helps produce um, the color production to see if there was any possible binding. So if there was a change in color, there was binding present from the of the phage. And finally, the stop solution is added to change the color one more time, and then the results are read through a plate reader at an absorbance of 450 nanometers. And finally, the results of the ELISA. This is an image of the 96 well plate after the experiment was conducted. The plate was organized in that the left half had the wild type M13 phage and the right half had the genetically modified SCFV phage. And the bottom row was conserved for a control full of just BSA blocking buffer. And as you descend down the plate, there is a decreased concentration of phage. So the most concentrated levels of phage were at the top and then it descends down. So looking at eye observation, it can be seen that the experiment that the modified SCFV phage has more binding than the wild type M13 phage. But looking over the results of the graph averages, here are the average phage graph binding to chitin from the ELISA. And it can be found that there is, for the SCFV phage, that there was an upward trend in the binding present, and it is found that the M13 phage varies at 1 e to the 10th virions or less, and that there is a present spike included at 1 e to the 8 virions, which requires further analyzation of the results, and generally an expected trend of what we wanted to see in the experiment. This is a graph for the binding to chitosan, and in this graph, it was present that there is a moderate amount of SCFV binding and that there is a peak at 0.5 at 5e to the 9th virions, and there's actually an opposite trend as the binding begins to decrease as the concentration increases, and that there for M13, there was slight binding that gradually begins to increase for chitosan. And finally, for the cellulose wells from the ELISA, there was a maximum binding level of the SCFV at 1 to the 8 virions at zero point 0.4, and that the M13 has interesting results, which varies in increasing and decreasing binding, and then begins to pan out. And overall, the same trend as with um, Pitocin, there was a decreasing trend as the concentration increases. So overall, the results have been summarized. For chitin, the more concentrated the SCFV phage was, the more binding, which was as expected. And then for all three carbohydrates, the M13 phage varied considerably. And for chitosan and cellulose, there was a moderate binding for the SCFV phage, which began to decrease in binding as the concentration increases, and finds a very interesting trend in how the binding occurred. Moreover, it can be summarized that SCFV had stronger binding levels and overall, and that the lower the concentration, the more binding as present in chitosan and cellulose. And there was a spike in all three carbohydrates for the SCFV BSA control. And moreover, chitin had the most successful binding in throughout the whole experiment, seeing the upwards trend in concentration increase. And chitosan and cellulose, as a result of the overlap in error bars, are more difficult to interpret. And the standard deviation calculations for each of the concentration of carbohydrate were higher than expected. And however, while the SCFB chitins error bars did overlap, the values were actually much closer to the expected numbers, which proves some successful results through this ELISA experiment. So going over to my discussion, the SCFB phage was expected to bind to chitin and chitin in the results, and cellulose was not expected to bind as successfully because it has a different chemical makeup of repeating glucose units. And moreover, that it can be determined that the genetic modifications to the SCFB phage did improve its receptivity to the carbohydrates overall. Moreover, um, even though M13, even though chitin is less water soluble than chitosan, the data is more indicative of the SCFB phage binding to chitin as a result of the pH levels, which I'll be going over to in the following slide. For future studies, there are some main takeaways to improve the flow of the experiment. One of them would be for the proper blocking of the wells, and this step was imperative for the BSA, in which we had to block the target antigen. Ensuring proper blocking is important, as it could lead to false positives if there was any surface of the plastic or of the well that was unblocked, and could lead to false positives and some of the ambiguous points that were on the graphs. So proper blocking with the BSA blocking buffer is important. Moreover, also to use a more acidic buffer is what was found to be could be more successful, as the polysaccharides that were used presented poor solubility in water and only dissolved in an aqueous acidic media. And since the NHCO3 buffer used had a pH of 8.6, it could have led to some insufficient coating of the plate, and as well as some false positives. So in the future, using a more acidic buffer would help the overall sticking of the target antigen to the bottom of the plate. And thirdly, running multiple trials would be helpful to acquire a larger data set and to be able to provide more trials to smooth out some of the errors that may have been presented in the experiment. Due to time constraints, only one trial of the ELISA was conducted, but in the future, running multiple trials would be more successful for the results. And finally, to conclude, the main points from the study and the ELISA find that the data is suggestive to chitin and proving um, promising results for the SCFV phage to bind this type of carbohydrate. However, some of the results, mainly in cellulose and chitosan, are inconclusive and require further study and analyzation to determine the effectiveness of the SCFV phage onto those two carbohydrates. And an overall further study of this one, the ELISA, and also phage therapy in general, as it has a very promising potential and a lot of and a very strong potential to be able to um, increase and improve the efficiency of phage therapy to help more patients in the healthcare setting. And finally, with increasing antibiotic resistant res- with increasing antibiotic resistance occurring overall globally, it's important that there should be more research in phage therapy to be able to be able to be able to I guess produce a more promising and a more effective treatment as bacteria is gaining more resistance to antibiotics, that phage therapy is able to take over and to be able to treat many patients in the healthcare setting. I want to give a special thank you to my mentor, Michelle V. Baxler, who has helped me throughout this summer and helped me open my eyes to the world of phage therapy, not really knowing much about it in the beginning of the summer, and now being able to have a presentation and write a research paper on it. I'm super grateful for for her helping me out over the summer, and to be able to conduct research in her lab has been an amazing and invaluable experience and opportunity, and I'm so grateful for that. Um, and that concludes my presentation. Thank you.
Yeah, I will stop recording.